Hello, everyone. Welcome back to Science Tweets with Pooja. I hope you all are doing well. So today our topic is components of living cell. Now you all know that living cell is basically made up of three parts. That is the outer boundary that we call as plasma membrane. Then we have cytoplasm and then we have nucleus. So we will understand various parts of living cell today, and specifically we will talk about plasma membrane in that. We will study about the structure of plasma membrane, then the most accepted model of the plasma membrane, that is fluid mosaic model. Then we will talk about various functions of plasma membrane, then what kind of transport happens across the plasma membrane, then what type of osmosis occur or what types of solutions are there. So we will be discussing one by one all of these points in our class today. So let's start quickly. So as you all can see on my screen that here plasma membrane cell is basically made up of three parts. Okay, that is your cell membrane, then we have this cytoplasm, and then we have nucleus. Now cell membrane, as our topic for today is cell membrane, the another name for cell membrane is plasma lemma, or we also call it as plasma membrane. As the name suggests, membrane, so it is basically the outer boundary of the cell. So what does it do? It separates the internal part of the cell from the external environment. Now, one of the a very important feature of plasma membrane that I would say is that it is semi-permeable, or you can say it is selectively permeable. Now, what do you mean by selectively permeable? Selectively permeable means it does not allow all the substances to go in or to go out, or it does not stop all the substances, but yes, certain substances are there, some specific substances are there which it allow to enter the cell or exit the cell. Okay, so this is one of the key feature of plasma membrane that I would say. Let us first of all talk about its structure. There were two scientists, that is Singer and Nicholson. They gave one model, although number of models were given earlier also, but the most accepted one is the fluid mosaic model. Now, as you all can see on my screen, what is this fluid mosaic model? Basically, it is basically that, can you see this round colored structure, yellow colored structure with two tail? This is lipids. These are lipids. So it has two lipid layer, upper one and the lower one. So this model suggests that our membrane, cell membrane, is made up of double phospholipid layer, and these are lipids basically, and these are polysaccharides. These are polysaccharides which are attached to the protein. So now what is this? These are protein. This pink colored structure that you can see in between the lipids, they are proteins, and some proteins you can see outside the lipids also. These are extrinsic proteins. So membrane has two types of protein: intrinsic proteins and extrinsic proteins. Intrinsic proteins are present in between the lipids, and extrinsic protein is present outside the lipids. That is, they are not present inside the membrane, or they are present on the periphery of the lipids. Okay, then we have cholesterol also in the plasma membrane. So this is the fluid mosaic model. Okay, now 75%, if I would tell you how much phospholipids is there, we have it 75%. 75% is the phospholipid, and rest is your protein, cholesterol, and polysaccharides. This structure you can see and you can understand easily. Now, as I told you that plasma membrane is selectively permeable, that means it will allow certain substances to come in and certain substances to go out. So it facilitates transport. What does it do? It facilitates transport. Now, you all know that transport means movement of molecules inside and outside the cell. Hmm? So basically, two types of transport occur over here. One is active transport and another is passive transport. So one is active and the other one is passive. Now, what is the difference between these two, active and passive? Now, see, active transport is happening very fast. It is a very fast transportation of molecules. Molecules like ions, okay, like, salts. All of these are transporting very fast at a very fast pace. And this requires a lot of energy as well. When amino acids, that is glucose, amino acids, ions, they are transported at a very fast pace, then that requires a lot of energy, and we call it as active transport. So on the contrary, if I talk about passive transport, so here passive means slow. So slow movement of molecules is going on. So here, no extra energy is needed. But yes, the condition is in what case this passive transport will happen, when there is concentration gradient. Now, what do you mean by this concentration gradient? Suppose outside the cell, suppose respiration is going on. Now, when respiration is going on, oxygen is being used up and lot of CO2 is produced. So when CO2 is produced, so inside the, inside the cell, CO2 is very high. Concentration of CO2 becomes very high inside the cell, and outside the cell, the concentration of oxygen is very high. Here, oxygen is very high. Now, since CO2 is very high inside as compared to outside, so movement of CO2 will happen from inside to outside. That means the CO2 will move out of the cell membrane. Whereas the concentration of oxygen is high outside the membrane. Okay, so oxygen will move inside the cell. Okay, so in this way exchange has happened, and this is due to the difference in their concentration. So it is happening slowly, continuously, and that is why we call it as passive transport. So two transports are very important, active transport and passive transport. Now, if we talk about passive transport, passive transport is further, can be classified into two types. Now, what are they? First of all, if the movement is of solute particles, agar solute particles move kar rahe hain, okay, so that will happen. Solute particles like gases hain, molecules, salts hain, amino acids hain, they all will move by diffusion. But if the movement of solvent molecule has to happen, suppose the exchange of water has to happen, water has to move from outside to inside the cell, then that will happen through osmosis. Very simple. So this is the basic difference. Now, yes, condition is again same. Movement will happen from high concentration to लो कॉन्सेंट्रेशन. तो अगर हम कह रहे हैं डिफ्यूजन, तो डिफ्यूजन में हम किसकी बात कर रहे हैं? सब्सटेंसेस की बात कर रहे हैं. वी आर टॉकिंग अबाउट सॉल्ट्स, आयंस, ग्लूकोज, अमीनो एसिड्स, all of them. If they has to come in, so their concentration needs to be higher outside the cell. So they will move, slowly and gradually, that is passively inside the cell. So they will move inside the cell by the process of diffusion. But suppose now water has to move, something in solvent state, it is in liquid state, that has to move, solvent molecules has to move. So they will move through this semi-permeable membrane by the process of osmosis. Am I clear? So osmosis is again very important, but the difference is that here movement of solvent molecules happen. Okay, so whenever you take lots of, during dehydration, whenever we feel dehydrated, so when we take lots of, liquids, so liquids, they ultimately go inside our cell and they hydrate our body. So that is by osmosis. So movement of solvent molecule is going on by osmosis. Now if I am talking about osmosis, now very important, let's talk about osmosis over here. अब देखिए, अगर मैं ऑस्मोसिस की बात कर रही हूं, सो ऑस्मोसिस में if I give you one cell, this is a cell, okay, and here water molecule, the concentration of water is high here outside, and inside it is less. So water will move from outside to inside. Okay, this type of osmosis is known as endo and osmosis. This is known as end osmosis. Very simple, means movement of solvent molecule is happening inside the cell. Whereas if in case you take a cell which is already very much, having high concentration of solvent molecule, now here the concentration is high inside the cell, so the cell will lose its solvent outside. So we will call this condition as X osmosis. Clear? Now, end osmosis, X osmosis. So that means the solution which is surrounding your cell can have varied concentration of solute and solvent. आपका जो सेल है वो जिस भी सोल्यूशन में है, उस सोल्यूशन का और आपकी सेल का जो सोल्यूट और सॉल्वेंट का कॉन्सेंट्रेशन है वो वेरी करेगा. So on the basis of varied concentration of solute and solvent, we can say that solutions can further be of 3 types. Solutions भी हम further classify कर सकते हैं. अब कैसे कर सकते हैं? अब देखिए, सबसे पहले हम बात करते हैं isotonic. What is this isotonic? Suppose कीजिए you have taken RBC आपने RBC cells लिए, ठीक है, and this is floating in 0.9% salt solution. 0.9% जो है concentration है salt solution की. So 0.9% salt solution concentration जो होता है, इसका मतलब क्या है, हमारे जो RBC का solute और solvent है concentration और जो बाहर जो solute और solvent का concentration, they are balanced. Okay, so when inside the cell and outside the cell solute and solvent are equal, then no exchange will happen. Do you feel that any exchange will happen? No, क्योंकि हमारा कंडीशन क्या है, ऑस्मोसिस है या डिफ्यूजन है, कंडीशन है कि दैट देयर हैज टू बी डिफरेंस इन द कॉन्सेंट्रेशन. तो अब यहां पर क्या होगा, क्योंकि दोनों साइड पे क्या है, इक्वल कॉन्सेंट्रेशन है, सो नो मूवमेंट ऑफ मॉलिक्यूल विल हैपन, सो इट इज आइसोटोनिक सोल्यूशन. तो 0.9% जो एक सॉल्ट सोल्यूशन होता है वो आइसोटोनिक सोल्यूशन होता है हमारे आरबीसीज के लिए. इफ सपोज आई टेक, let us say 0.7% salt solution. अगर मैंने कितना salt solution ले लिया, 0.7% salt solution ले लिया, उस केस में क्या होगा, देखो आपका जो RBC है और बाहर जो salt solution है, बाहर का जो solution है उसका solute लेस है और उसका solvent ज्यादा है. 0.9 पर बैलेंस्ड था, इसका मतलब आरबीसी के अंदर 0.9% इतना तो सॉल्ट होता ही होता है. अब 0.7 का मतलब क्या हो गया, 0.7 का मतलब हो गया बाहर हमने सॉल्ट के कंसेंट्रेशन को क्या कर दिया, रिड्यूस कर दिया. बाहर हमारा सॉल्ट रिड्यूस हो गया, मतलब हमारा सोल्यूट रिड्यूस हो गया एंड सॉल्वेंट इज मोर, आपका जो सॉल्वेंट है वो बाहर ज्यादा है, सॉल्वेंट इज मोर. सो व्हेन सॉल्वेंट इज मोर आउटसाइड, तो क्या होगा, this solvent will move inside the cell. That means end osmosis will happen. That means swelling up of cell will happen. ऐसे solution को we call as hypotonic solution. इसको हम बोलते हैं hypotonic solution. Okay. Hypotonic means एक ऐसा solution जिसकी solute concentration was less but solvent concentration was high as compared to the concentration of our cell. जो हमारा सेल है उसके अंदर जो सॉल्ट और सॉल्यूट और सॉल्वेंट है उनके कॉन्सेंट्रेशंस का हमने कंपैरिजन करके बता दिया. अब सपोज करो मैंने ले लिया 0.13%, 0.13% सॉल्ट सोल्यूशन. 0.13 का मतलब क्या हो गया, आपने सॉल्ट बहुत हाई ले लिया, सोल्यूट इज वेरी हाई. हियर सॉल्ट इज वेरी हाई और सॉल्वेंट कैसा है, सॉल्ट बाहर हाई है तो अंदर कम है, सॉल्वेंट बाहर लेस है तो अंदर ज्यादा है. सो दैट मीन्स water will move out. That means X osmosis will occur. And when salt concentration is very high, we call such solutions as hypertonic solution. So, three types of solutions: isotonic, hypotonic, hypertonic, and in all the three cases, our cell is behaving differently. Isotonic, no exchange of solvent will happen here. Hypotonic, yes, because the salt concentration is less, solvent is very high, so it will go inside the cell. That means endosmosis will happen. Hypertonic means salt is very high. Hyper means high, means salt is very high, solute is very high. When solute is very high, then what will happen? The solvent is less. That means X osmosis will happen over here. Okay, so this was all about this topic. So I hope you have understood, and I would like to know more topics that you want to study. So you are most welcome to write in the comment box about the topics that you want me to make videos upon. Thank you so much. Bye-bye, and please take care.